Truly remarkable, Professor. Thank you. Last but not least, it is time for us to embark on a journey through the self-regenerative capacities of peripheral nerves. So as our final speaker of the panel, in her research at the 3B Center in Braga, she has focused on finding an easier way for nerves to reconstruct themselves, utilizing for that purpose a scaffold of sorts to guide their growth. These are the nerve guidance conduits by Dr. Cristiana Carvalho. Please welcome her.
Hello. Good afternoon, everyone. I hope you're all listening to me and you're able to look at my presentation. So first of all, I would like to acknowledge the other speakers for their... Sorry, I think we have a problem with the presentation. Okay, so I think I can start now. First of all, I'd like to thank for this invitation to participate in this amazing conference and seminar. I would like to acknowledge all of the other speakers for their very interesting presentation, and thank you all of you who are home in these strange times watching via internet. So my name is Cristiana. I'm a postdoc researcher at 3B's research group, and today I'll be talking to you about biomaterials for the development of conduits for guided peripheral nerve regeneration. But first of all, I'll make a brief introduction to you about how peripheral nerve works and why we should focus on its repair and regeneration. So, as you can see in this video, we have the nervous system composed of the central nervous system and the peripheral nervous system widely spread throughout our body. The central nervous system is composed by the brain and spinal cord and protected by bone. On the other hand, the peripheral nervous system functions like a highway where the information travels from the outside and the environment to the central nervous system, or the other way around, through afferent and afferent neurons. But why should we focus on the repair of this system? Because 1 in 1,000 individuals are affected by these injuries every year, and this is associated to a high economical burden. It is estimated that $150 billion healthcare expenses per year are spent in this kind of injuries, and because the treatments that we have nowadays are not effective, individuals do not return to work at least 1.5 years after the intervention. So the injuries that usually occur in the peripheral nervous system are like this, represented in this schematic. Usually there is a gap or a discontinuation of the nerve. So we need a surgical repair. The optimal surgical repair should be the end-to-end suture, where you suture the nerves back together, but that can only be done if it is done tension-free. When that is not possible, we move to the second option, which is the autologous nerve graft. Basically, you pick up a piece of a nerve of any other part of your body and you use it in your injury. It is considered the gold standard. However, it is associated to several drawbacks. As you can see, you end up with a new lesion at the donor site. There's often a size mismatch, allodynia, which is some kind of neurological pain, and the recovery is often seldom. And the third option that we are here to talk about and to discuss are the engineered nerve guidance conduits. So basically, when you have a gap or a discontinuity in your nerve, the technique mostly used is tubulization. So you use a conduit to protect your injury, and that stimulates the regeneration. There are, up to this day, several FDA-approved hollow nerve guidance conduits. However, they present poor outcomes. So in this table, you can find the FDA-approved nerve guidance conduits that are used in clinical setting nowadays, but they all have the same kind of restraint. They are empty conduits, which means they lack both topographical and chemical cues. So this is the reason why they underperform when compared to the autograft in terms of both motor and sensory recovery. And this is where tissue engineering and regenerative medicine strategies can help us overcome this problem. So I believe, it is my belief, that tissue engineering can contribute to solve this problem of peripheral nerve regeneration and the lack of good outcomes. How? So basically, the tubulization itself, besides the use of tubular conduit to protect your injury, tissue engineering can contribute in a lot of ways. You can engineer your conduit to have an inner structure that will provide topographical guiding and topographical cue. You can also add transplanted cells, extracellular matrix, and neurotrophic factors. There are neurological factors that will stimulate neuronal regeneration. During my research, I have worked basically with natural biomaterials. Some are mentioned here, and today we'll focus mainly on gel and gum and silk fiber. But why are good materials a good option to try to solve this problem? Mainly because of their high abundance, which makes them fairly cheap, because of their biodegradability, biocompatibility, which means they do not usually elicit an immune response when implanted in human body, and high tunability, which means you can modify them according to your needs. So these are the advantages. And as an example, I'll talk to you about a work developed by me and my team focused on the use of gel and gum. So many of you might not know what gel and gum is. Gel and gum is a polysaccharide obtained from a bacteria that behaves very similarly to gelatin. It gelifies when increasing the temperature. So we decided in a tissue engineering advanced strategy to try to use this gel and gum as a luminal filler inside our nerve guidance conduit to try to add some topographical cues and try to enhance nerve regeneration. So at this point in the beginning of the work, our hypothesis was, is it beneficial to use freeze-dried gel and gum in the lumen of the conduits? So that was our task, and we have several types of gel and gum. So we have high-acyl gel and gum, low-acyl gel and gum, a form of metacrilated gel and gum, and according to all of them have different properties, they behave differently, so different formulations with different amounts of these two compounds will give rise to different scaffolds that behave differently. And our goal was to discover which of these formulations would behave best. So the first step was to produce the scaffolds. This down here is a 3D reconstruction of the obtained scaffold, just as we wanted, a very porous one. And as a preliminary in vitro test, we seeded Schwann cells on these scaffolds. Why Schwann cells? Because Schwann cells are the main cells of the peripheral nervous system, and they are the cells that mostly contribute to a regenerative process. And what you can see here, these green dots, are live cells. So you can see that as we move from this corner to this corner, we increase the amount of metacrilated gel and gum in the formulations, and we have an increased number of cells, an increased metabolic activity, which means the cells are liking this new environment of metacrilated gel and gum. So this is a hint of what we can expect in vivo. The next step will be to produce the conduits to really implant them in animal. So we prepare them by injecting the different formulations inside the conduits. And again, you can see a 3D reconstruction of our developed conduit. It's a chitosan conduit outside with our freeze-dried porous structure of gel and gum inside. So then we implant it in an animal model in mice. What we do is cut 10 millimeters of the nerve of the rat, of the sciatic nerve of the rat, and implant our conduits. And we do a battery of tests. To start, histological evaluation is often the basis of the study, and now we initiate the study. And what you can see here, especially after 12 weeks, is that the formulation that contains more metacrilated gel and gum is the one that allowed to obtain a more robust nerve cable that regenerated throughout the 10 millimeters that we cut. So, in one hand, we have the in vitro results pointing to the fact that metacrilated gel and gum would perform better. Histological evaluation also pointed to that direction. In the electrophysiological results we obtained, again, we had better results in the same formulation, pointing to the metacrilated gel and gum being the best formulation, which again happened when we performed histology to assess the vascularization process. And at this point, I need to tell you that vascularization, or the angiogenesis process, is extremely important in terms of neuronal regeneration, because it is an indicator. The more vascularized is your tissue, the more robust will nerve regeneration be. So the more vascularization you have, the more successful you can be in your outcomes. So as conclusions of this work, we proved that inside all of the gel and gum formulations that we have, the ones that contain higher amounts of related gel and gum proved to be the right material for further studies and to continue this study. And well, it was the first time that gel and gum, this kind of polymer, was used ever for peripheral nerve applications. And now, switching the biomaterial, we can move from gel and gum to silk fibrin. And yes, silk fibrin is exactly what you're thinking, is the one used in textiles and produced by the silkworms. And me and my team were able in the lab to develop a brand new processing method to fabricate an enzymatically cross-linked silk fibrin nerve guidance conduit. And it was brand new, that it was even patented. And we were very happy with the preliminary results. As you can see in these videos, they were very preliminary, but they seem promising because they are suture friendly, which means they can withstand a suture being pulled out of them. They are easy to handle, kinking resistance, and present shape recovery. So these are just some preliminary videos, but of course, when you develop a technology for the first time, you have to thoroughly characterize it. And the permeability is very important and will dictate whether your nerve guidance conduit will be successful or not. Because your conduit needs to be permeable enough for the permeation of nutrients and oxygen, your regenerating nerves, but on the other hand, it cannot be porous enough to allow cellular infiltration. So after a battery of tests where we assessed molecular permeability and cellular permeability, we got the results that we were hoping, which are that the conduits are 100% permeable to molecules such as nutrients and oxygen, because we need them inside to get in contact with our nerve, but on the other hand, they are impermeable to cellular infiltration because we don't need any other cells or components there in the place where regeneration is taking place. And of course, we also had to study the biological properties of our brand new developed conduits. We tested them in vitro. Again, we just put some Schwann cells on it. Is what you can see are these in green Schwann cells that seem to adhere and proliferate well. It means this is a good matrix for Schwann cell invasion and proliferation. These are good news. And then when tested in vivo, they were subcutaneously implanted in mice, and after 8 weeks, we explanted them, and we can see they are well tolerated by the host. There's no immune response. They are well vascularized and well integrated within the tissue. So we were able to develop a brand new fabrication method regarding silk fibrin nerve guidance conduits. And I want you to notice, these two videos down here, where there's a comparison between a commercially available chitosan conduit called the Axon when you kink, what happens is that the conduit bends. And imagine you have your nerve inside here, it will be occluded and smashed and damaged. On the other hand, in our developed silk fibrin conduits, when you bend them, they are completely kinking resistant, which means that your nerve will be intact inside the conduit. So this was the process of developing the conduit itself. But now again, we want to use a more complex tissue engineering strategy to try to enhance our chances of nerve regeneration. And we wanted to add, at this time, neurotrophic factors to our silk fibrin conduits. And that is exactly what we did. So based on the literature, we chose these two growth factors, NGF and GDNF, because they act upon different neurons. And there is a body of evidence that confirms that the exogenous support of these growth factors enhances the survival and the proliferation of neurons. So, given the exact same fabrication process that I showed you previously, the enzymatically cross-linked silk fibrin nerve guidance conduit, we just have to find ways of incorporating our growth factors, which we did. We incorporated growth factors in two different ways. The first, we call it the cross-linking method, where we envision the growth factors would be cross-linked and very well entrapped in the walls of conduit, suggesting the slow and low release of the growth factors. And as a second method, we have the absorption method, where we envision a faster release of the growth factors from the conduit to the medium. And we have to study exactly that, release kinetics of our growth factor. And we set up this study. So basically you put the conduit in culture medium and let it release to the medium, and then you analyze your medium to quantify the amount of growth factor that was released. And as you can see by the graphs, and as we predicted, the cross-linking method significantly decreased the release of the neurotrophic factors, because they are so well entrapped in the walls. And on the other hand, the absorption method allow a higher release of the growth factors. And now that we know how the growth factor is released, we still have to prove that these growth factors that are released to the medium are still bioactive and will have and perform as they should. So we did a biological assay, culturing dorsal root ganglia with our medium. What is our dorsal root ganglia? There are some structures you have near your spine, which are the direct interface between the central nervous system and the peripheral nervous system. So they are the perfect model to study, to perform this kind of studies, and because they have neurites that respond to stimulus. And so by testing all of our formulations, we could see that this formulation containing GDNF with the method of absorption was the one that allowed us to have a dorsal root ganglia with the highest neurite elongation, but also the highest neurite density. And so, taking advantage of that, we chose this formulation to move on to an in vivo assay using a 10 millimeters sciatic nerve defect in rat model. So our study groups are our conduit containing GDNF, a plain silk fibrin conduit with no growth factor at all, and our positive control, the autograft. So there are different techniques, different approaches that you can use to assess how regeneration is going. We did a bunch of them. We did histology. We did percentage of muscle weight loss, which confirms our GDNF was acting upon the motor neurons. We also performed pinch tests, which confirms that released GDNF was acting upon sensory neurons. And we were also able, through histological analysis, to assess axonal regeneration and vascularization. So as you can see here in this picture, you see these round white circles are actually axons. So by counting them, you can assess the axonal regeneration. And here, of course, you can see the blood vessels. Again, the higher the vascularization, the higher are the chances for peripheral nerve regeneration. generation. So after studying all of these parameters and correlating all of them together, we could get to the conclusion that GBNF released at the injury site by our nerve guidance conduit seems to be exerting a protective effect in the axons and leading to a higher vascularization and higher nerve regeneration. So we were successful in this tissue engineering approach of introducing the growth factors in our silk fibrin conduits, and they, in fact, as we hoped, performed significantly better in all parameters evaluated when compared to the plain silk fibrin conduits. So what I want to transmit to you as general conclusions is that the options that we have available in the market, and if you have in fact an injury and if you go to the hospital, the options that you get are not satisfactory enough. So we as scientists have the obligation to use tissue engineering approaches such as adding luminal fillers or discovering new methods to produce nerve guidance conduits or adding the growth factors, and try to, in a combinatorial approach, try to get these technologies together to try to improve the outcomes as much as possible, because today we are not happy with the technologies we have available in terms of peripheral nerve regeneration. And so this is all for me. I would like to thank you for your attention. And thank you all the partners and funding for supporting this work. And these are my colleagues at this research group. So thank you.